Good afternoon. My name is Amorce Lima. I'm a clinical scientist at the Esoteric Research Lab at Tampa General Hospital. Just a little bit about the Esoteric Testing Lab. This lab is being led by Dr. Susan Silbert, who is here with us. And Dr. Ray Wyden was the one who pioneered that lab. And we have three sub-departments from the esoteric testing lab. One is the molecular testing, who takes care of all this clinical sample testing, providing results to the hospital. And there's also the flow lab, and we are the research. There's a research part of the esoteric testing lab, and that sub-department has 6 different people in it. I am included, so we do a lot of clinical trial, and we also provide residual specimen for sponsors who want to do in-house testing for assay development. So we've done a lot of tests, validation as well. So that's enough for all the advertisement, but we were very pleased and to be able to be involved in the analytical and clinical performance evaluation of the CMV Quant assay using plasma and non-plasma samples. So this is what we will be presenting to you as a data today. If you live in the Tampa Bay area, you would hear that a lot, the Tampa General Hospital, how the hospitals practice medicine, and we define it. So since our new CEO had taken over a little bit 4 years ago, he had done a lot to actually lead the hospital the right direction, and a lot has been done since. So our vision is to be the safest and most innovative academic center in America, and this is the big why that we have as the hospital. And what do we do exactly? We heal, we teach, we innovate, we care for everyone every day. And how do we do that? We are a big hospital of over 1,000 beds with over 8,000 employees. We are the primary teaching hospital for USF Health, Boston College of Medicine. We are recognized for our world-class care. We are named the #1 hospital in Tampa Bay and top 10 in the nation for patient care in different categories such as cancer care, neurology, neurosurgery, pulmonology, and lung surgery. We also have other specialties such as internal medicine, cardiovascular, orthopedics, endocrinology, as well as Children's Medical Center. We are known to be one of the leading and busiest transplant program in the United States. So this table here gives you the number of solid organ transplant that we done the year of 2021 last year, and you can see the number of transplant that we've done was 553 solid organ transplant. Most of them were kidney transplant with 330, and liver transplant almost 150. So we are ranked number 3 in the nation based on volume for kidney transplant, both for adults and pediatrics. Organ transplant is basically life-saving treatment for patients with end-stage organ failure. Statistics have shown that at least 20 people die each day waiting for a transplant. Fortunately, there are good news, which last year had shown that there were at least 41,000 transplants done, which is a 6% increase from the previous year. This is the first time there's more than 40,000 transplants done, solid organ transplant done, and that shows you there are more people volunteering and donate organ to be transplanted. And those numbers were shown that the kidney and heart and liver transplant there were record numbers of those solid organ transplant done last year, and also there were record numbers as well of disease organ donation for those transplant. However, there's still challenges that our patients are still faced with, and one of them is long-term care outcome uncertainty. as well as post-transplant infection that may occur and organ failure. So there are a lot of viruses that are associated with post-transplant infection. Some of the common ones are in the herpes group, as Karen just mentioned, cytomegalovirus is one, a big one, EBV, herpes simplex virus 1, 2, the group of polyoma virus, BK and JC, hepatitis virus B and C, and as well as the respiratory viruses. So as a lab serving that big hospital, we ought to be able to test for all those different viral targets and also providing those results to the hospital as soon as possible. So those numbers here show you the number of tests that we have for those different targets. CMV, we've done over 500 per month, and those numbers reflect what we have done the past 6 months. But because of COVID, we don't know how that we know a few years ago, some of those numbers were even higher. So the pandemic might have affected some of the numbers from the past to what we're seeing right now. But nevertheless, we are a big transplant hospital, and we have to be able to test those and provide the results to the hospital. So 250 SSV1 and 2, EBV 150, and VZV 60, and no virus 20. Those are the average numbers of those targets that we've tested monthly. So how far have we gone? So we know as a lab, we in the past, it seems like it's been many months ago, but we have gone from manual extraction, manual convention, PCR, amplification, and using gel electrophoresis, as well as enzyme-like hybridization to be able to detect those targets. Move from there to automated system for extraction, still manual PCR, having to set up PCR reagent and put on the thermocycler using the Hologic or the simplexer, and to more of an automated system where we can get sample to answer results. So that have enabled us to move from 4 or 5 days before we could get results to 4 hours and perhaps now less than 3 hours. And also a lot of hands-on time to now minimal hands-on time. Knowing the importance of CMV and all this post-transplant infection, we were pleased to be able to partner with Hologic to do this validation, where the goal was to evaluate the CMV assay, Quant assay for detection of CMV on the Panther automated system, and get the data and compare it to the standard of care, which is the ELITech MGB alert CMV at ASR on the ELITech Ingenious automated system. So full disclaimer, the protocol was submitted to the IRB, was approved to use residual specimen, and this protocol was followed, as well as good clinical practice was used and other requirements pertinent to the in vitro molecular testing devices. The methodology used for the study, first we did the analytical performance, where we used the SeraCare AccuTrack CMV DNA quantification panel to measure the accuracy of the assay. The linearity was also assessed using the exact diagnostic, the CMV verification panel, with different concentration from 2.6 log IU per ml to 6.6 log IU per ml. Limit of detection was assessed as well, where exact diagnostic CMV panel was used, and 5 replicates of several dilutions were used were tested to determine the limit of detection of the assay. This data here shows the linearity between the Aptima CMV in log IU per ml versus what was expected. You can see there's a straight, nice line here with perfect slope and an R squared of 99.9945. And the same was seen as well for the ELITech CMV versus the expected values. The accuracy was assessed where we have the Aptima, the graph on your left, the Aptima CMV in IU per ml versus the expected. Again, pretty nice slope, showing good correlation between the Aptima CMV assay versus what was expected. And the same can be seen as well for the engineered CMV assay versus the expected log from the hierarchy tract panel. The accuracy table is showing here. We put this data on this table so you could see here we have on your left, the far left, the different samples, the expected and RU paramel, the ELITech CMV expected values, the Aptima CMV expected values, and the difference between the expected for ELITech and the expected, and then the difference in their for the Aptima and expected, and then the difference between the Aptima and ELITech values. So what I'd like for you to look is at the mean difference on the bottom, where it is showing that the mean difference between the ELITech and expected is close to 0.6 log IU per ml, and the mean difference between the Aptima and expected is 0.14, and the Aptima and the difference and the mean difference between Aptima and ELITech is almost 0.5 log. So just keep in mind, because those values, we'll get back to them once we talk about the clinical samples. That is showing that ELITech is quantitating those samples at 0.6 log higher than what is expected, and also they are quanting the ELITech assays quanting those samples close to 0.5 log higher than what we're seeing from the Aptima assay. So for the limit of detection, different dilutions were one, as I mentioned, in 5 replicates, in the several replicates, and we are seeing here the way that the assay was designed was to test for 20 different replicates and on both Aptima and ELITech. Turns out that the dilution was done in Aptima buffer, and the ELITech instrument did not like the dilution in the Aptima buffer, so we had to redo the LOD for the Aptima using water, nucleus-free water. So that's why you see there's a discrepancy in terms of the number of replicates for the Aptima assay versus the ELITech assay. But in all, what we have seen was that the limit of detection for the Aptima was 50 IU per ml, and for the ELITech, it was 200 IU per ml. But we know this is what the result is showing here based on this study. But previous studies have shown that previous validation have shown that the limit of detection for the ELITech CMV assay is in between 50 to 100 IU per ml. For the clinical performance, we used residual specimen, as I mentioned earlier. 249 samples were tested. 141 of those samples were frozen. They were tested first on the standard of care assay, which is the ELITech, and then they were frozen to be run on the using the Aptima CMV assay. And also there were 108 fresh samples that were used in this validation. They were tested right after they were tested on the standard of care. Of those samples, 217 were plasma samples, and 32 were different other samples, non-plasma samples, such as respiratory samples and GI and congenital samples. So the results showed that out of the 217 samples, 134 of them were positive for both assay, and 66 of them were negative for both assays. And we saw some discrepant samples. There were 17 of them, and all of the samples that were discrepant, they were around the lower limit of quantification of each of the assays. Out of those 17 samples, majority of them were detected by the Aptima CMV assay as opposed to the ELITech assay. And you could see the positive PPA, the positive percent agreement between the two assays, and negative percent agreement between the assays as well. For the non-plasma samples, there were 32 of them, and all of them, 26 of them were detected positive for both assays, and 6 were negative for both assays. So there were 100% agreement between both assays for the non-plasma samples. The clinical, we wanted to look at the clinical correlation between the two assays. As we can see on the left, we have the dimming regression showing how both assays perform versus each other. You have Aptima on the y-axis and ELITech on the x-axis, showing there's a pretty good slope of 1 between them, and R square of 0.88, still a good R square. But we wanted to look further the correlation between the two assays, which is given by the blend and output plot. And we, again, showing the blend output plot on the y-axis is plotted the difference between the Aptima and the ELITech CMV assay, and the x-axis giving you the average of all the samples between the numbers, the quant for each of the samples, for each of the assays. And what this is showing now is showing that there is the bias is 0.5 log IU per ml. This is again showing not only in the analytical performance, but also in the clinical performance that the ELITech is quanting those samples at 0.5, at least 0.5 IU per ml higher than the Aptima. Advantage and the disadvantages between the two assays. We know the Aptima CMV assay on the Hologic Panther. The Panther it's a random access instrument, but assay menu, we can run different assays on the instrument. We don't have to wait for the run to be done and load new tests as we have to do for the Ingenious instrument, which require a batch analyzer to actually fully taking advantage of the reagent. And for the Hologic Panther, the reagent coming ready to use, as well as the controls and minimal hands-on time is needed, versus the Ingenious instrument where reagent has to be prepped and made. The Hologic Panther is much faster, also showing, giving you higher throughput, in which you can run 36 samples in less than 3 hours, versus the Ingenious, where the fact that you have to wait for one to be over before you put other ones, it takes much longer, therefore a slower turnaround time. The only disadvantages that we can name for Hologic Panther is although 500 microliter of sample is needed, but you're required to put 700 microliter of samples in there. And the 200 that's left over, you can actually remove it and use it for other purpose if needs be, versus the Ingenious instrument where you only need 200 microliters of samples. In conclusion, we saw a good, very good overall agreement between the two assays. Although there were, as I mentioned, there were some discrepancies, but those discrepancies were in lower limit of quantification, and then again the analytical performance showed very good performance by the Aptima CMV assay on the Panther, with 50 IU per ml as the limit of detection. And again, one thing that to keep in mind is the Aptima CMV assay is on average 0.5 log IU per ml lower than what we're seeing from the ELITech MGB ASR assay. Then I just mentioned the benefit where the Panther is random access, reagent coming ready to use, and you end up having minimal hands-on time and higher throughput on the Panther versus the Ingenious instrument. So just wanted to thank the people from the lab, special thanks to Vicky and Laura, who worked on the assay and doing the validation. Again, we have the name of all the people in the lab, so I just wanted to thank them, and thanks Hologic for inviting me to present the research, the validation results that we have done. Thank you.